Hello and welcome to another episode. I'm pretty sure a lot of you who are actually my subscribers must be wondering why I've been teaching a lot of garbage topics, transport mechanisms, vesicular transport, the normal stuff, active transport and all that, the structure of the membrane, glycocalyx, all rubbish things that are not that important for the exam. Okay? Today onwards, you're going to understand why I made sure to do all that first. Okay, so first off, we'll start with GIT absorption, and you'll understand why all the transport mechanisms that I was talking about will make it so much more easier for you. So if you haven't already watched all those videos, go check them out first, because I'm not going to spend a lot of time explaining more in depth about the transport mechanism because I've already covered it. Okay, in case I wanted to do that, this lecture would go on for 1 to 2 hours if I tried explaining everything. Okay, so I'm going to go ahead with this to make you understand how important it is to understand the basic transport mechanisms and help you understand GIT absorption. Okay, so in case you're again wondering why on earth do I need to even know about GIT absorption, take a look at this. Lactase deficiency, your lactose intolerance that happens because of it, celiac disease, glucose galactose malabsorption, heart nerves, lysinuric protein intolerance, pernicious anemia, cystinuria. In Crohn's disease, what happens post ileal resection, and steatorrhea. These are just very few examples. There are lots more to this. Okay, so understanding this topic helps you understand a lot about all these diseases, how they happen, how you can treat it, everything. And that is very essential to become a doctor. Okay, so let's just make a quick game for you in case while you're doing this. Uh, Comment below how whatever I'm teaching you today is related to all these diseases and how it is relevant. Okay, so first we'll talk about your enterocyte. This is the luminal side, this is the basolateral side, and you have your basic sodium potassium ATPase, which is the primary active transport. Okay, so because of this, in the basolateral segment, sodium is going outside, sodium is lesser inside, so that now creates a gradient for sodium to come inside. So sodium coming inside, along with it, it drags something like amino acids, glucose, galactose, lactose. Okay, so this is a sort of second reactive transport. This is a co-transport. Okay, so that is how all these things are absorbed: amino acids, glucose, galactose, lactose. What about water? Okay, so we already noticed how here sodium is more in the basolateral segments outside. Okay, so now that itself has created osmotic gradient over here. So through the paracellular, so this was transcellular, through the cell, through the paracellular route, through the osmotic gradient, so what is going to go through osmosis? Okay, and this is regulated by your tight junctions. Okay, so how easily this can pass through, that is dependent basically upon the tightness of your tight junction. So this was sodium, excess sodium here. Okay, so a positively charged ion, to maintain electroneutrality, a negative ion should go in. That again passes through the paracellular route because of the whole thing that is there. So along with the water, chloride also goes this side. Apart from this, chloride has another way. Chloride gets exchanged with bicarbonate. So this is again something you see very often in your protein. Okay, anion exchange between chloride and bicarbonate. So this takes place in the ileum and the large intestine. So it's like the distal parts. And the reason for this is, from the bacteria, metabolism, all that will result in acid production, and to neutralize that acidity, this mechanism is there so that your alkaline bicarbonate is secreted in the distal parts like the ileum and large intestine. Okay, so just remember the major part is like this. There is also this person. So is bicarbonate only secreted like this? No. So in the initial part, bicarbonate has to be absorbed. So for bicarbonate to get absorbed, that is where your non-ionic diffusion is coming into picture. Okay, so already we've seen that because of sodium potassium ATPase, sodium is lesser here. So it's easier for sodium to come in. Sodium comes in, H plus goes out. Okay, so this will result in formation of H2CO3. H2CO3 splits into CO2, H2O, and the CO2, because it is lipid soluble, can directly pass through the membrane. So this is your non-ionic diffusion. Okay, so by this you can see almost all type of transport here. The primary active transport, the secondary active transport here. You can see the osmosis here. You can see passive diffusion here, and you can see symporters here, co-transport, counter-transport. Okay, and you can see non-ionic diffusion here. Okay, so we already mentioned glucose, galactose, and lactose through secondary active. What remaining is your fructose. So fructose is something that goes through facilitated diffusion. So there again, an example of facilitated diffusion. So now that is done, we'll go into a little more depth about carbohydrate transport. Okay, so in your enterocyte, you have your glucose, galactose, lactose. This goes through your sodium co-transport system. Okay, so this is your SGLT1. So this is your sodium glucose transporter 1, and through that glucose, galactose, lactose goes in. So this is an example of a secondary active transporter. Fructose, as I had mentioned earlier, goes through GLUT5, and this is facilitated diffusion. Okay. After this, the glucose, galactose, lactose, as well as fructose, all of this goes out through the basolateral segment through GLUT2. Okay. So fructose, once it goes inside, either it can go and get converted into glucose, or it can directly go out. So in either way, it can go out eventually as fructose itself, or get converted to glucose. Okay, so this is an example of secondary active transport. This is an example of a facilitated diffusion, and these both are all facilitated diffusion. So a lot of questions can come interchangeably from rearranging this. So this is pretty important. Now we'll come to proteins, peptides, amino acids, and see how they are absorbed. Okay, so first thing is the intact proteins as such. So this is kind of important in the postnatal period, where your immunoglobulins from the mother through the breast milk is absorbed by the infant. Okay, later on it's not much there, but very few percentage of intact proteins, even in adults, are absorbed through this. Okay, and all this is pretty much in the distal parts, not in the initial parts of the intestine. Okay, so this happens through your endocytosis, and in the endocytosis, it's a clathrin mediated endocytosis. Again, clathrin, I explained how it happens. Okay, so in case you don't know, go check out that video. So clathrin mediated endocytosis, it goes into endosome, multivascular bodies are formed, lysosome degradation, and finally it goes out into the circulation, and it's been absorbed like this. So that was your intact protein. Now we'll go on to oligopeptides. Okay, so these proteins are further degraded and they become peptides. Okay, further degraded, they become your amino acids. So we'll first talk about the peptides. So this oligopeptide hasn't been completely degraded into amino acids yet. Okay, so your sodium potassium ATPase is there. Because of that, sodium is lesser here, sodium comes in, H plus goes out. Now instead of your sodium dependent co-transport, this is a proton dependent co-transport. Okay, so this is a peptide transporter 1, pep2. Okay, so H plus comes in, and along with that your oligopeptide comes. So this is an example of a secondary active transport. Okay, so peptide comes in, and once inside, the peptide is acted upon by peptidase and it forms amino acids. Again, it can go out through the basolateral segment. Okay, so this is one example why, even though there are certain deficiencies in amino acid transporters, you don't actually manifest much symptoms unless it is much severe or some acute setting where other environmental factors exacerbate it. That is because even though that deficiency is there, through this mechanism it can still reabsorb things. So in cystinuria, in Hartnupp's disease and all that, you notice that you can still not end up having manifestations because of this. Okay. And once amino acids are finally formed from your digestion process, that gets absorbed. Okay, so just know that for amino acids, there are like 7 transporters in the luminal segment. Okay, out of the 7 transporters, 5 of them are sodium dependent co-transports. Out of these 5, 2 also have chloride involvement as well. So along with sodium, chloride is also involved in the transport. Okay, and then we have 2 things which are independent of sodium. So one example is your hydrogen that I was talking about. So all of these are ultimately co-transporters, secondary active co-transporters. Okay, so that is how amino acids come inside. Next is your basolateral segment and how it finally goes out. Okay, so there again sodium, potassium, ATPase. Sodium is more outside, sodium is less inside. So this forms a mechanism for sodium to come in and your amino acids are going out. And this along with it might not only be there, there will be involvement of other ions as well. Okay, chloride, hydrogen, all can be involved. Okay, so I'm not going too much into detail of this, which is not needed for you guys. So that is there. Apart from that, through facilitated diffusion, amino acids can go out. Okay, so there are 2 mechanisms, either a sodium dependent mechanism, sodium independent mechanisms are as also there. Okay, apart from all this, amino acids can be taken back inside. Yes, because enterocytes also require nutrition. Okay, so they are taken back inside as well. Okay, so this, what you have to understand is there are 2 types of, you have your villus cells as well as intestinal crypt cells. Villus cells are those which absorb, crypt cells are those which secrete. So villus cells, they absorb and take it out into the circulation. So there, all these other mechanisms are more active. Okay, when you talk about intestinal crypt cells where secretion is taking place, there they need these amino acids to come back into it. So that is a little more prominent in intestinal crypt cells. Okay, further things I have not included in this beautiful diagram along with a table in Boron's textbook. So in case you want to read a little more, you can read that. Okay, otherwise it's not needed. It's actually given in renal system over there. So at your level, I don't think it's needed to go that much in deep. Okay. So next is the lipids. So when you talk about lipids, this is where you need to understand the difference between your polar structure, non-polar things, okay, which is lipid soluble, which is water soluble, and what is the nature of your membrane. All that helps in understanding this. Okay, so we have your lipids which are there. Lipids are not water soluble. Okay, so what you have to understand is it needs to get transported through your intestinal secretions and all that. Okay, so the whole chyme is there, it has to go there, and to reach the surface of the villus for absorption, it has to go through an unstirred layer. So in the center there is a stirred layer, but as you go closer towards the luminal part, which is having the villi and all that, you notice that next to it there's an unstirred layer of fluid which is there. So this has to pass through that. So that is why it needs what is called the micelle. Okay, so bile salt has 2 sides, it's just like phospholipid actually, like in your membrane. Okay, so one side is polar, one side is non-polar. So it helps attach one part to your lipid, other part to the water outside. So this helps transport it like this. Okay, so once all the fatty acids, your triglycerides, and cholesterol esters, all this, okay, uh, once they move inside, okay, and in this whole process digestion takes place. Okay, the digestion process inside takes place, converting them. Okay, so they move in, digestion takes place, and as they go, the end products like fatty acids, okay, so they start coming out. So for that to come out, that is when this acidic environment over here helps. So again, we had seen how sodium goes in because sodium potassium ATPase is there, and H plus goes out. This acidification kind of helps bring out these fatty acids from here outside, because right now they are lipid soluble. Okay, so this lipid structures, once they get protonated like that, and that will help them shift out from this phase inside. Okay, because their lipid solubility is what is making them stay there. So that is shifted out, and then once they're here, through various transporters they are taken in. And some of them, it's not all through transporters. Along with transporters, what is noticed is that it can even go directly. Okay, so some of the lipids which are actually staying as lipid soluble, they can go through. Others get transported through the channels that are there. Okay, so there are various ways by which they are taken inside once they are inside. So once they are inside, they need to go into the circulation. Okay, so this is again another set of steps. So this is where chylomicron is coming into the picture. So earlier was micelle to help them cross through and get in. So once inside, they form chylomicron or VLDL and come out. Okay, so what you have to understand is we have short chain fatty acid, medium chain fatty acids. So they don't need this whole process. Okay, short chain fatty acids are not that water repellent. Okay, so they can actually pass through this directly, but they have also lipid solubility. So they basically go through directly by diffusion and then go directly out also into the blood. Okay, so this is very less in butterfat and all that, or in the later part of the intestine from your colonic bacteria, short chain fatty acids are produced. So these come on directly like this and go out directly into the blood circulation. The other ones with micelle that come in, once they come inside, they get reesterified. Okay, so you have all these fatty acids, monoacyl, glycerol, lysophospholipid, all this get converted to cholesterol ester, phospholipids, triacylglycerol, and they are coated with apolipoprotein protein structure by the endoplasmic reticulum and packed by the Golgi apparatus, and finally they are going out, so that again they can go through circulation in any way. So this usually goes through the lymphatic circulation. Okay, this is why the whole process is taking place. Okay, so there's a lot of endocytic processes. Understanding all the mechanisms actually helps. Okay, so this is like a perfect example to understand how different transport mechanisms that we have talked about till now are helpful to understand this whole picture. Okay, if you don't know any of that, I'm not even sure if you understood what I just mentioned. Okay, so that is your lipid. Next we'll come to vitamins. Okay, so I know you'll be tired by now. Okay, so you just take a break. You can pause it and watch it later. Or you can just stretch. Okay, anyone who's actually been in my offline classes would know what I would ask you guys to do by now. Yeah, I'll just make everyone jump up and actually literally jump up and stretch all over the class, and then I'll resume it. Okay, so now once we're fresh again, let's come back to it. Vitamins. We have your fat soluble vitamin, water soluble vitamin. The fat soluble vitamin, whatever. Just now we were talking about lipid absorption. So in the same process, along with the micelle, chylomicron, it just goes the same way. Okay. The other things are the water soluble ones. So majority of the water soluble ones go through a sodium co-transporter mechanism. Okay. Second reactor, sodium co-transporter. The exceptions are your vitamin B12 and folate. So in folate, what you notice is that this is a special one. Okay, unlike what we usually see. So folate comes in, and OH minus, your hydroxyl, is exchanged for that. So this sort of exchange mechanism with hydroxyl is involved for absorption of folate. Okay, now we'll go to vitamin B12. Okay, so cyano cobalamin. So just remember that this fat soluble, water soluble, folate, all this are in the upper part of the intestine. Vitamin B12 is much distal. Okay, so cobalamin is coming. It reaches the stomach, and in the stomach, it binds with your haptocorin to form a complex. Okay, also remember that intrinsic factor is secreted by the parietal cells, the same cells that secrete acid. So intrinsic factor is secreted. Okay, now your pancreatic secretion. So this has reached your duodenum. Your pancreatic secretions are going to come, and they're going to result in degradation of your haptocorin, proteolytic, right? So the haptocorin is degraded, and that releases your cobalamin now. Now cobalamin is transported by complexing with your intrinsic factor. So remember this, although intrinsic factor is secreted in the stomach, intrinsic factor and cobalamin binds together only after your proteolytic degradation of haptocorin by your pancreatic enzymes. Okay, and then it is transported, and as it is transported, it reaches your ileum. So this whole thing is finally absorbed in the ileum. So if you remember what I had shown you earlier, upper intestine is where all the other vitamins are absorbed, but this vitamin B12 goes to the distal parts, ileum. Okay, now in the ileum we have the enterocytes. A receptor mediated endocytosis takes place and it goes in. Okay, so now lysosome acts and lysosomal degradation, and your cobalamin is finally coming out of the vesicle. Okay, so that is where you have to know transcobalamin. So the whole concept now is for cobalamin to go into circulation and get carried, but for that it needs a transporter. So that is your transcobalamin 2. Okay, so now lysosomal destruction, and it comes out and complexes with your transcobalamin 2 and is carried into the circulation. So that is essentially how your vitamin B12 is absorbed. So just remember that ileum is where this happens, because a lot of MCQ questions come from this. Okay. Next is your minerals. So we have a calcium. We have your divalent metal transporters. So most important example of that is your iron. Then we have magnesium. Okay. So calcium divalent metal transporters, all that absorption takes place in your duodenum, upper part. Magnesium, through an active transport, it is absorbed from your ileum. Okay, but some passive mechanisms also in the rest of it. So magnesium is not completely fully understood. Okay, we will not go into detail of that. Just know that in the ileum, active transport is there. Rest of the intestine, a passive absorption is there. Okay, duodenum is where calcium and your iron is absorbed. So we'll go into detail of calcium and your divalent metal transporters. So when you talk about calcium, calcium is absorbed through your TRPV6 channels. Okay, so calcium comes in like that through these channels. This is purely based on your diffusion gradient. Okay, so there's nothing much into it. There's no exchanger, import and all that. Okay, calcium is less inside, so calcium comes in. It's pretty much that what happens in your body. Okay, calcium is less inside the body, inside the cells. Okay, so calcium comes in. Now after coming in, the calcium binds with cal binding, and for it to go out, there are two mechanisms. One mechanism is an exchanger. Sodium comes in and your calcium goes out. So this is one mechanism by which calcium can go out. Another thing is, by the use of ATP, your H plus can come in and calcium can go out. Okay, so here ATP is not required because your sodium potassium ATP is already made sure that sodium is lesser here. Okay, here H plus is more here. Okay, it's always more acidic inside the cell, so you need ATP to move H plus inside. Now how does vitamin D come into the whole picture? So vitamin D acts on its nuclear receptor over here, and it results in transcription, translation, and the whole protein synthesis. One thing it does is increased. These channels are increased, TRPV6. So increased absorption. Okay, cal binding is increased. So more calcium can bind to cal binding. And also the transcription translation of these basolateral mechanism that we just saw. Both of them are increased. So this is how your vitamin D plays a role in absorption of calcium. Next is your divalent metal transporter. So that is for your iron. So this I'm not going to go too much into detail because this again I'm going to make a separate video because this is very important. A lot of concepts here. Okay, so I'll make it short over here. There are two sources of iron. Okay, your heme iron, non-heme iron. Heme iron, heme along with a hydrogen symporter like this, heme is going to come, and then inside heme is getting destroyed and Fe2 plus is released. Or you have your Fe3 plus, which is the source, non-heme ion. Okay, so this through either in the stomach, it gets converted through to Fe2 plus. Okay, so that takes place by in the presence of your acid in the stomach. So either that happens, and if that doesn't happen also before absorption, your duodenal cytochrome B activity will make it convert Fe3 plus to Fe2 plus. So this is a divalent, right? This is a divalent transporter. So a trivalent thing cannot come. So divalent metal transporter. So this again through H plus as importer. Okay, so H plus, how it becomes high outside, you already know. I don't have to explain this again. Sodium potassium ATPase and maybe a sodium hydrogen exchanger might be there, and that will result in the whole mechanism. Okay, so that comes in like that, and after that it gets stored as ferritin. So again for that it gets stored as Fe3 plus inside, and then again if it goes out, it has to go as Fe2 plus in a transporter called the ferroportin. Okay, and but if it needs to go in the circulation, it has to combine with your transferrin. For that it needs to be Fe3 plus. So that is where something called the hephaestin is there. Okay, so that converts Fe2 plus to Fe3 plus. So to go inside, it needs to be Fe2 plus. Okay, to get stored as ferritin, Fe3 plus. To go out again, it needs to be Fe2 plus. And after going out to actually bind to transferrin for circulation, it needs to become Fe3 plus again. Okay, I hope this is not too confusing. Don't worry, I'll put a separate video out for this. Okay, so coming to the gist of it, this is what you have to understand. Okay, iron, calcium, folate, all you can see the absorption is predominantly in the duodenum. Okay, calcium is also absorbed in other parts also. When you talk about bile acid, cobalamin, these were two things that we had seen earlier was in the ileum, distal part of the ileum. Okay, so just remember this: bile acid, cobalamin, distal part of the ileum. Most of the other things are in the upper parts. Okay, take a look at this. You can see that sugar, amino acid, water soluble fat, water soluble and fat soluble vitamin, long chain fatty acid, everything is predominantly in the upper parts. Okay, your all the ions, okay, all the ions were increasingly in your upper parts. Okay, you can see that bile acid, vitamin B12, and if you remember, I was talking about the receptor mediated endocytosis process in newborns where antibodies from the mother's milk, okay, so intact protein, so that also takes place in the lower part. Okay, also remember potassium is secreted in colon and all that. Okay, so sodium and potassium are always going to be opposite. Sodium potassium ATP, so that resulted in increased potassium inside. So that is a mechanism where potassium gets secreted eventually. Okay, so this happens in the colon. Okay, so I hope you understood this. Okay, this was a very brief description to make you understand how studying about transport mechanism will help you a great deal in understanding a lot of topics. Okay, later on I will put up more videos to explain in detail each of these things I told. Okay, so don't worry about that. Okay, so this was basically to help generalize and understand how transport mechanisms are going to help. Okay, so this will help you out in a long way. And the next thing, apart from this, I'll be uploading videos on how transport mechanism is going to help you understand acid-base balance, how it's going to help you understand your renal transport mechanisms as well. Okay, so stay tuned in. Thank you.